Well, one potential reason relates to nitrogen and phosphorus excretion rates. So nitrogen and phosphorus excretion scales with body size, such that the same biomass of fish, if that biomass is represented by a lot of small fish, are going to actually excrete nitrogen and phosphorus at a higher rate than if that biomass is held within a few large fish. Okay, so we would now expect that high predation populations might have elevated nutrient turnover rates, right, or be driving higher nutrient turnover rates. In fact, when we measure excretion rates for the two different population types, we see this effect. We see that for nitrogen and for phosphorus, we see high predation populations have higher excretion rates per hour than do low predation populations. Okay, so this is potentially an effect whereby guppies are increasing nutrient availability for algal growth, right, by recycling nitrogen and phosphorus and making that available for algal production. Okay, so now we have sort of armed with this suite of possible trait differences and ecological differences, we can now start to investigate whether or not those things truly do have important ecological effects on communities and ecosystems. And here we ran a mesocosm experiment in these stream mesocosms. Some of you in the room are familiar with these stream mesocosms. And into these mesocosms, we placed either high predation guppies plus rivulus, low predation guppies plus rivulus, or rivulus only. And you'll notice there's rivulus in all these mesocosms, and that's because in natural streams, guppies always coexist with rivulus. So a system that didn't have rivulus in it, in my mind, wouldn't represent the true variation we see in nature. They always coexist with rivulus, so we decided to include rivulus in these treatments. And then instead of a no-fish treatment like we had in the alewife system, we have a rivulus-only treatment, where we replace the biomass of guppies with the biomass of rivulus, right? So the biomass of fish in all of these treatments is the same. Okay, so we measure a suite of community and ecosystem response variables. Here I'm just going to talk about benthic invertebrate biomass and algal biomass. And the important thing to take away here is simply that low predation guppies seem to drive especially decreases in algal biomass relative to high predation guppies. Okay, we see a slight difference in benthic invertebrate biomass, but that's not a significant difference. Interestingly, the rivulus-only treatment falls between those two guppy treatments. Okay, so that's interesting because it appears that the guppy type you introduce to the stream determines which direction the system goes, right? So if you were to introduce high predation guppies to a rivulus-only stream, you would expect to see an increase in algal biomass. Whereas if you introduce low predation guppies to a rivulus-only stream, you would expect to see a decrease in algal biomass. Okay, that is certainly an unexpected result if you're an ecologist that tends to believe that a guppy is a guppy. This fundamentally suggests that the type of guppy you introduce might push the system in two totally different directions, okay? And so if you're thinking about this sort of in the context of the trophic cascade framework that I set up for the alewife system, we can think about the effect that predators might first have on guppy density, right? So predators would tend to decrease guppy density, which might tend to increase algal biomass. But then, as you'll recall, consumption rates for high predation guppies are lower, right? So this decreased consumption rate might actually magnify this difference and cause an even stronger cascade. And then you'll recall that excretion rates are higher for high predation guppies, right? So this fertilization effect, driven by nitrogen and phosphorus turnover, might further amplify this trophic cascade, such that at the end of the day, we've seen the ecological cascade here having been amplified either up or down, modified by these other mechanisms that are related to the trait differences between the populations themselves. Okay? All right, so similarly to alewife, where we had in less than 50 generations, you can see dramatic differences in ecological effects. We can see the same thing in guppies, right? In as few as 20 generations, you can see that high predation versus low predation guppies either drive an increase or a decrease in algal biomass. Okay? All right, so I want to come back now to the first two questions that I posed at the beginning. So the first one, does intraspecific consumer biodiversity have important ecological effects? And I think the studies in these two systems show that it's possible, that it can. That actually different populations might perform differently, might feed differently, might excrete nutrients differently, and that those can potentially be manifest in communities and ecosystems in ways that we can measure in controlled experiments. The second question is a critical one though, and that is how do these intraspecific effects measure up to species level effects, okay? We as people interested in ecoevolutionary dynamics, specifically interested in intraspecific diversity, we might sit around and talk all day. Andrew and I might talk all day in our offices about how cool this is, but it's unlikely that an ecologist is going to pay much attention to us unless we can show that these effects measure up compared to something that they're already interested in and that they already recognize as having a strong effect, okay? So the next stage of this work was to think about how these effects measure up. And the first thing I did was to simply look at the effect sizes for these two experiments that I was involved in, okay? So look at the effect sizes across all the response variables we measured for guppies, all the response variables we measured for alewife, and compare the effect of the presence or the absence of alewife or the presence or absence of guppy, right, to this contemporary trait change, low predation guppy versus high predation guppy, or landlocked alewife versus anadromous alewife. You can see in the guppy experiment, we have 1, 2, 3 cases out of 5 where this contemporary trait change effect actually was larger in magnitude than the guppy presence absence effect. And in the alewife examples, we have 1, 2, 3, 4 out of 6 examples where the difference between anadromous and landlocked alewife was actually larger in its effect size than the presence or absence of alewife. Okay? So this started to now make me think whether these are peculiar study systems or whether these are general features of systems that had essentially been overlooked, right? Because differences between guppy populations are not really that obvious, right? Or differences between alewife populations are not all that obvious. They look like alewives in a lake, they look like guppies in a stream, but they seem to be functioning quite differently. So what do other experiments say? Okay, so this part of the talk is what we began talking about at this QCBS working group. So we started to try to pull together a suite of other experimental studies asking these same questions and see how consistent those, the results from those studies were. What time is this? What time do we go till?
I don't know. I mean, I don't know the first part. What time is it?
It's quarters. So
Yeah,
We're 15 more minutes. Okay, thanks. Okay, so first I'll talk about experimental approaches, and essentially we use 2 types of experiments. We use removal experiments, which are similar to what I talked about in the alewife experiments, right? So where we compare life history variation, in this case, landlocked compared to anadromous, and contrast that with the removal of that species from the system, okay? The second type of experiment we looked at is the kind we ran in the guppy mesocosms, okay? We have LP guppies versus HP guppies, and in the no guppy treatment, we didn't remove fish entirely, we replaced the biomass of guppies with equal biomass of rivulets. So we consider that a replacement, okay? And that's a somewhat important distinction to keep in mind, because in cases where we have the removal, it could represent the removal of something like an entire trophic level from that study, whereas in a replacement, oftentimes you have the replacement of one sort of species with another, but that species might be at the same trophic level, okay? So we might expect effect sizes to be somewhat larger for these kind of experiments compared to these. We wanted to recognize potentially those differences, Okay, so as we went through searching for different studies, we came up with some. There aren't an overwhelming number of studies, at least at the time that we did this meta-analysis. There's more coming out all the time, actually. And if we collected a new bunch of studies today, we could probably get twice as many experiments, but this is what we had at the time, right? So we have my experiment, an alewife, and then our lake comparisons. This Post et al. Study was essentially the similar suite of response variables, but instead of being measured in mesocosms, these were measured in the actual lakes themselves. We have the Guppy experiment, so we have my experiment, right, where we measured benthic invertebrates, periphyton decomposition, nutrient excretion. The Basser experiment that came out relatively soon after my experiment and measured the response variables that we measured plus a suite of additional ecosystem level response variables. And of course, there's more of these coming also. We found a suite of Daphnia studies. Some of these look at differences among clones. This one here is interesting in that it actually looks at trait differences in Daphnia that are driven actually by these alewife populations. So actually, it's an evo cascade, right? Because we've got the the effect of trait divergence in alewife causes trait divergence in Daphnia, causes some ecological effect, right? Net primary production. And then there's a study of Daphnia that's related to, this one is related to standing genetic variation. This one is specifically related to evolution of Daphnia in response to toxic cyanobacteria and their efficiency at consuming phytoplankton. We have a couple of studies performed in stickleback, mesocosm studies, and this study that was done in pale chub. And this one is an interesting study in particular because all of these studies represent differences among populations, whereas this study actually looks at different feeding types within one population. Okay, so here we have population level differences. Here we have intra-population level differences in that study. Okay, so we looked at all the response variables from this suite of experiments. We calculated effect sizes. Essentially, we compared intraspecific effects, which are basically the types, right? Landlocked versus anadromous, or high predation versus low predation, and compared that to the effect of either species removal or replacement. So anadromous plus landlocked compared to no alewife. Okay, assuming that basically life history type doesn't matter, right? Population of origin doesn't matter. Their presence is what matters, okay? And in this case, if we see a positive value of Hedges G, we get a stronger intraspecific effect, and if we get a negative value, we get a larger species effect, okay? What we're really interested in is kind of this, this point at which, which is bigger. Of course, our expectation is that species effect should be bigger, right? The differences among species are bigger than the differences within species, and the removal of a species or a trophic level should have a bigger effect, right? That's our sort of our thinking going into this, right? So if we look at the distribution of Hedges G across all these different studies and all these different response variables, okay? Here's our sort of zero, right? So at this point, intraspecific effects are of the same magnitude as species effects. So you can see, first of all, that the most frequent observation was that species effects are slightly larger than intraspecific effects, okay? But we have a reasonable number of cases where actually intraspecific effects are bigger than species level effects, right? And we have some cases where species effects are bigger, not surprisingly. If we look at what systems are represented in each of these categories, we can see all the study systems for some response variables. All the study systems represented across those experiments showed some response variables being affected more strongly by species effects, okay? This here is this study, right? So intra-population variation, at least on this tiny sample size, seems to be the weakest form of within species variation, okay? If we look here at intraspecific effects, we see these three systems show up on this side: alewife, guppy, and Daphnia. Okay? We know alewife has already been called a keystone species, right? So maybe something about that keystone role is important. Daphnia, we know, has big effects on phytoplankton abundance, right? So there could be something about the ecological role of a species that determines whether or not variation within that species makes a big difference. Also, interestingly, we broke down these response variables into either community level or ecosystem level responses, okay? And so there's a little bit of ambiguity here, right? Some response variables could arguably be called either community or ecosystem. We did the best we could at categorizing things, but the result here was actually quite surprising and interesting. For what we called community level response variables, we see stronger species effects. For what we called ecosystem level response variables, we actually see the intraspecific effects being of equal magnitude to the species effects. Okay? And here, for communities, we had things like zooplankton community, benthic invertebrate community. For intraspecific effects, we have things like primary production, decomposition, nutrient cycling. Those response variables seem to be strongly affected by intraspecific variation, okay? This was a pretty unexpected result. We didn't really have an a priori expectation for which way this might go. But it makes a little bit of sense in the sense that some of these response variables are being impacted by both the consumption and the nutrient excretion, right? So we might have this sort of amplified cascade or these synergistic effects being more prevalent here than down here. But generally speaking, why we see this, we're still not sure. How real is this pattern? Will it stand up if we include more studies? We're not sure. It's a sort of an interesting hypothesis. And one of the ideas that I had was, well, why have these effects been overlooked for a long time? It could be because community ecologists are frequently those that are interested in species effects and biodiversity effects. And if it's true that for most community response variables, species presence or absence or species identity is the most important thing, maybe that's a good reason why community ecologists have focused on those things and not paid a lot of attention to intraspecific variation. So why is this important? So actually Andrew and I and Mike Kinnison and a few others put together this paper in which we mapped human-induced trait change, all the cases that were available at this time. And there's a lot of cases, and probably this is, certainly only the places that people have looked for trait change are represented on this map. There's a lot of places where people probably haven't looked, but where humans are driving contemporary trait changes equally as they are in North America or in Australia and New Zealand. And so if we're driving these dramatic changes, rapid changes in the traits of wild organisms, and if the potential exists for these trait changes to be manifest as dramatic changes to community and ecosystems, this sort of provides an impetus for us to better understand what kind of effects generally trait changes might have. Right? It's a general phenomenon. Really, if we're going to try to understand global change and the ecology of global change, these sorts of phenotypic variation and phenotypic change through time, I would argue, are part of the equation that we should not be ignoring. Okay, so in conclusion, intraspecific effects are important for species with key ecological roles. Okay, we showed that in alewife, or dramatic phenotypic variation, for example, in guppy, okay? Intraspecific effects can be as large as species effects. We see that in both alewife and guppy and across a larger suite of systems. And that these effects tend to be large, especially for these ecosystem level response variables. Although we're not sure why that is, and we don't know how that result will hold up when more studies are included. Intraspecific effects may be especially important when consumption and excretion act synergistically. Okay, so this conclusion is one that is drawn from these aquatic study systems, but it also could be applicable to terrestrial systems where nutrient cycling and consumption are also important. If those things push systems in the same direction, we might see these synergistic effects manifest as large ecosystem and community effects. And then I would speculate that higher rates of human-induced trait change, therefore, might be reshaping ecosystems worldwide. And then if we're interested in global change, that not just those trait changes themselves, but if we're interested in ecological changes, widespread ecological changes, we should be thinking about these trait changes and how they are translated into ecological changes. With that, I'd like to thank all the co-authors on the work that I presented, QCBS for funding, NSF, a few other funding sources, and thanks for your attention.